Welcome everybody, whoever everybody is. Hopefully we have some people from the Brazilian Reproducibility Initiative, some from outside. You're all welcome here. We're pleased to have Malcolm Macleod as a speaker today. I'll present him in a couple of minutes, but I'll start with some announcements, which I think are important. So let me just do this. Yes. First of all, if you're participating lab in the Brazilian Reproducibility Initiative and you have not sent us information on the experiments you can perform and the animals you can use, please do so now. I mean, there's like, I don't know, half a dozen people missing still. So, I mean, maybe none of them are here. So, I mean, it could not be you, but we really need this. I mean, we've been asking this for a while now. We have selected the experiments for the initiative already. We have to divide them across groups in the country at the end of this week. We should be doing it on Thursday and Friday, and it's actually quite a hard process. So ideally, we'd like to do it like with everybody's data. If you have to like redo it next week, it's going to take us a lot of time. So please send us information. One, on the experiments you feel confident about performing from the ones we sent you. Second, the kind of animals you use and the kind of animals you can receive. So those are two questions you have been asked by email. So please reply if you have not yet. Second thing, this actually goes for everyone interested. It goes for our labs, but it also goes for like whoever is interested. We just opened up the registration, the pre-registration for the survey of beliefs in prediction markets part of the initiative. So in this, you actually have to guess or bet, and you actually get money to bet on that. But the idea is to whether you can predict which experiments will be successfully replicated. You can sign up at this Google Forms. You can find this link in our Facebook page as well. The markets and surveys should be happening around June or probably July, most likely. But you can already register now. It's important for us to get a feeling of how many people we get. For more information on that, you can watch Pedro's talk on our first meeting. That's on YouTube at the link which is there. Or you can also show up next month as our speaker for this webinar on the 4th of June will be Anna Dreber from Stockholm, who's actually a big expert in prediction markets in science, who's been helping us along in doing this. So she's responsible for like most of the prediction markets in other replication initiatives. Final call, if you're a student or a postdoc in Brazil, we're holding a hack week in July. It's the No Budget Science Hack Week. It's a sister project to the Brazilian Reproducibility Initiative. We're trying to discuss ways to improve science, both locally and globally. We had a debate a couple of weeks ago. Should be putting that on. You can find that in the Facebook page, but we should be putting an edited version online soon. On bringing up people, so like what could be interesting projects. A lot of teams have come along. These are them, and we have a website online already. So the idea will be to have like a mix between a hackathon and a summer course. So a 1 week course with tools, with lectures, and with project development. So people developing projects in groups of 4 or 5. We do have a website which is under the Brazilian Reproducibility Initiative website. So it's reprodutibilidade.br/No Budget Science Hack Week. We should start registration soon as well. So registration should open like next week or the following one. We're starting in May. We'll hold that open until June. The Hack Week is in the end of July, from the 22nd to the 28th. And we're welcoming young people from around Brazil to work in projects to improve science in the biomedical field and as a whole. And this is what I had for announcements. So after this, I'm pleased to introduce Malcolm, who will be our lecturer for today. I have to unshare my screen now. So Malcolm is a good friend from Edinburgh. He's been at this for a much longer time than we have. I mean, he's been trying to really build initiatives to improve science for maybe a couple of decades now, or something like that. And he's the founder and coordinator of CAMARADES, who is a network of researchers dedicated to meta research and research synthesis, meta-analysis in preclinical science, which is, I think, a very, very big avenue for developing a more robust science. He also does a lot of work in terms of how can we make clinical and basic research be more free of bias, more reliable, and I think he's going to share some thoughts on that. And he's collaborated with us. He's actually a big part of why we do this. I mean, when we were actually starting on this meta science business, I think that was like 5 years ago. That was the year we brought Malcolm in, and that somehow helped our group get a spurt and like actually make this a meaningful part of what we do. And these days is pretty much everything we do in the lab. So we're very grateful for Malcolm to have collaborated with us. We're very grateful for him to be with us today. So yeah, up to you.
Well, thank you very much, Olavo. I'm pleased to hear that I'm responsible for everything that you're doing, presumably because you're trying not to make the same mistakes that I did. So hello, everybody. My name is Malcolm Macleod. I'm joining you from Edinburgh in Scotland today. I always forget at the end of my talks to thank my collaborators. So there was an art installation that won one of our British prizes, the Turner Prize. It was a Tracy Emin. It was an unmade bed with condom wrappers and underwear. It was called All the People I've Ever Slept With. So this is at least some of the people that I've collaborated with, because I'll forget to thank them later. Apart from this lady here, Lindsay Thompson, who I have collaborated and written with, but I'm also married to, and she's the only one that falls into both categories. I've got some conflicts, but so everyone who comes to tell you a story, to try and tell you something, has got some conflicts. I sit on the British drugs regulators, also for the time being the European neurology drug regulator, although that may change. Marcus Manaffa and Chris Chambers and colleagues have set up the UK Reproducibility Network, and I'm involved in the steering group with that. An independent statistical standing committee for a Huntington's disease charity. I coordinate the Equipped Innovative Medicines Initiative, where we're trying to create a quality framework for in vivo research. So when I do some in vivo research and I want to convince someone, I've got, if you like, a badge or a way of demonstrating to them that I did it properly. And I don't know if anybody's seen the blogs about the minimum standards framework for reporting biomedical research that Nature and Science and Cell and others are putting together, growing out of the top initiative, and I'm involved in the group that's developing that, and hopefully we'll get that launched later this year. So let me see if I can get my slide. So like everything in life, the more I do this stuff, the less I really understand that I know. Because when I started off, I didn't even have to think about what research was, because everyone knows what research is. Research is just what you do, is what researchers do. But as I get older, I find it more and more important to try and set out exactly what it is that I'm thinking. So for me, research stems from the analysis and interpretation of observations. The setting of those observations may be spontaneous or experimental, and spontaneous observations we call observational research, and experimental observations we call hypothesis testing research. And that distinction is important, and often we get that distinction wrong, where we think that things are hypothesis testing, experimental studies aren't really. And that research usually leads to a knowledge claim, and that knowledge claim usually involves a statistical analysis. And for me, it's very difficult to do, if you like, due diligence to assess the provenance of a knowledge claim unless you understand the statistical analysis, whether the research was done in a spontaneous setting or experimental setting, the nature of how the research was planned, designed, conducted, and analyzed. And so this brings us then to the problem of reproducibility and replication. And I think it's useful near the beginning to set out some definitions. The diagram here is borrowed from Steve Goodman and John Ioannidis and Dan Finelli had a piece in Science Translational Medicine some years ago where they talked about reproducibility of findings and reproducibility of experimental findings and robustness and the like. But initially, reproducibility related to the reanalysis of the same data set following the same analytical procedures. And that started off in computer science. I try and write this code, you try and write this code, we put the same thing in, do we get the same thing out at the end? And replication was held to require the collection of new data following the same methods. So our problem here is that we talk about a reproducibility crisis in biomedicine, but actually, do we mean a replication crisis? Or what I mean is this issue of reproducibility of experimental findings. So me and my lab sets out to design an experiment of this type with this design to test X, and I collect some data and analyze it. And then someone else comes along and tries to follow the same experimental design in their lab. Do they get the same results, or do they get different results? And non-scientific audiences think this is self-evident. Of course, you get the same results because these are facts that come out of experiments. Well, we've got some experience now from replication studies as to whether that's the case or not. To start off with, these were retrospective studies that were done largely by the pharmaceutical industry. They were retrospective, where pharma companies shared their historical experience where they'd attempted replication. So they'd read something in the literature, they'd been at SFN and they'd seen something as they walked down one of the poster aisles, and they said, we could maybe do something with that. And then the first thing they do is they bring it in-house and try and replicate it. And when Bayer did that, they could only replicate 1/3 of 67 findings over 4 years. When Amgen tried it, Glenn Begley, they could replicate 1 in 9 studies. And Glenn's experience at Amgen is instructive, because for 5 of the findings that they really thought were pivotal, and these were actually in vitro findings, they went back to the originator labs and said, look, we'd really like to make something of this, but we've tried to do the experiment 6 times and we still don't get the answer that you got. And one of the guys said, yeah, that's funny. We had to do it 8 times before we got the answer that's in the paper. But you get the answer once, it must be true. And it's not true. But there's a problem with these retrospective replication studies. So first of all, there's possibly a selection bias. That's to say, we've got 2 companies have published their experience, but that's 2 companies out of, I don't know, 100, 200, 300 pharma companies around the world. If only 2 of them have a problem and 298 don't, then there isn't a crisis. And then, of course, the other thing is recall bias, because these studies weren't set up ex ante, but someone's gone to the files and they've looked through the files to try and find everything they can. And some of those compounds or some of those hypotheses that were brought in may be missing from the files, and they may be missing from the files because they were completely rubbish, or they were maybe missing from the files because they're completely brilliant. So we don't really know how good these retrospective replication studies are at telling us the scale of the problem or the prevalence of the problem, but it was sufficient to convince most people that there was indeed a problem. And then we come to prospective replication studies. So these are different. These are academic-led. Great attention is given to faithfulness to the original study design, adequate statistical power, and many of these studies have got statistical powers which are 2, 3, 4-fold greater than the original study. Pre-registration, working with the authors of the original studies to try and make sure everything, the study materials and the likes, just right. And we've now had at least 4 of these. Although I found out the other day there's also a replication study in experimental philosophy, whatever experimental philosophy is. What we see in them, though, is fairly consistent, that the rate of replication is between 36% and 62%. So roughly half, give or take 10% of studies will replicate and half won't. And then at the edge here, we've got... I need to take off my Apple Watch because it's trying to answer my questions for me. And then if we look at those studies which did replicate, so this isn't just all the studies, these are in the studies where they found a statistically significant effect in the same direction as the effect in the original study. What was the effect size observed? The effect size observed is about half of what it was in the originator study. So across the range here, across a variety of research domains, there are problems with taking findings in the literature which we think are true and showing that you can get the same thing. Now, of course, there are problems potentially with this, with selection bias. So how did the originators of these studies choose what findings they were going to attempt to replicate? Because if you were one of the psychology methodological terrorist gang, and that's what they've been accused of, wouldn't you deliberately go around selecting all those psychology studies that you knew were false because they would show your replication study in good light, because you'd find a whole lot of things that didn't replicate? So there hasn't been random sampling of studies to replicate. in social science replication study, the most recent CAMARADES one, they deliberately selected social science studies published in Science or Nature. Now, I find it difficult to get well-designed hypothesis testing biomedical research into Science or Nature, not because it's not good, but because they don't think it's newsworthy enough. To get something in the social sciences into Science or Nature, it's going to be astonishingly newsworthy. And to be astonishingly newsworthy usually means that the findings aren't true, as I'll show in a minute. So, that 62% I think is very good, much higher than I would have expected for replication of that kind of study. But I think we're now at the stage where we can make a general claim that the lack of reproducibility of experimental findings has been observed across such a wide variety of research domains that it can be considered to be a general phenomenon rather than specific to individual research domains. And that therefore, unless a field can demonstrate that they don't have a replication problem, it's reasonable for us to assume that they do. And what we're seeing now is wave and wave and wave of replication reproducibility studies in different research domains. And I'm sure that eventually we'll find some research domains where there's good reproducibility, and maybe that's PCRs or Affymetrix gene chips or something. Maybe, we're bound to find something that's reliably reproducible, but until we know what's reproducible and what's not, it's difficult to work out what parts of our literatures we can believe and exploit and which parts we should leave alone. And that's important because one of the major challenges in research is translating findings from the research domain in which they occurred into more widespread usage. So you could imagine about taking a finding from in vitro research and moving it into animals, or taking research from animals and moving it into human clinical trial. This is a road sign from Wales, and it says on it, no entry for heavy goods vehicles, residential site only. And because apparently there might be some sheep or lorry drivers in Wales who don't read English, they've also written the same thing underneath in their particular minority language in Welsh. Except what that actually says in Welsh is, I am not in the office at the moment, send any work to be translated. So if you like, this translational road signist hasn't understood the black box of their data analysis. They've taken their text string and they've sent it off to get processed, and they've got the answer back, and they've said, oh, well, that must be what it is. We'll put that on the road sign, and they look pretty stupid. We're scientists. We would never do anything stupid like that. I apologize to those of you who've already been introduced to the Atlantic salmon. This is a technique called functional MRI imaging. It relies on the basis that when you use part of your brain, you increase the metabolic requirement for oxygen and the metabolic production of waste products, and that causes a reflex vasodilatation that can be detected in something called the blood oxygen level dependent signal. And so it's a kind of modern day phrenology. You put someone in a scanner, you ask them to perform a certain task, and the relevant part of the brain will light up and say, this is. These machines are very temperamental. They need to be tended and loved and looked after and set up every week to perform well. And this group in the States had a competition to see who could bring in the most unusual piece of organic material to put in the scanner on a Monday morning to get it all set up for the week. And one day Craig is on his way to work and he remembers that it's his turn for something for the scanner. And he'd forgotten something to put in, but fortunately his route to work took him past the Boston Fish Market. He thought no one's had a fish in the scanner before, might make it smell for a day or two, but I'll win this year's prize. And so they put the fish in the scanner, they twiddled the knob, they got it all set up to go. And then what happened was that their healthy human volunteer on the Monday afternoon didn't turn up. And Craig's genius was to do the study on the salmon. So one mature Atlantic salmon participated in the fMRI study. It was 18 inches long, it weighed 3.8 pounds, and they didn't know what sex it was. And the task administered to the salmon involved completing an open-ended mentalizing task. So the salmon was shown a series of photographs of human subjects with a specified emotional valence. They were happy or sad or perplexed. And the salmon was asked to determine which emotion the human in the photograph was experiencing at the time. They identified several active voxels within the salmon brain cavity with a cluster level significance of less than 1 in 1,000. And as they conclude, either we stumbled onto a rather amazing discovery in terms of post-mortem ichthyological cognition, That's to say dead salmon can think, or there's something wrong with this whole approach. And of course, there's something wrong with the approach as it was enabled then, and for which they received the Ig Nobel Prize for Neuroscience in 2012, and rightly so. But of course, we are proper scientists, aren't we? We would never do anything like this. This is a treatment close to my heart, experimental focal ischemia with low-dose glutamate and homeopathic Arnica montana. And here you see that half-dose low-dose glutamate improves things slightly, and full-dose low-dose glutamate improves things by 40%, which is the kind of industry standard for a neuroprotective drug. But when I tell you that half-dose low-dose glutamate is 10 to the minus 120 molar and full dose is 10 to the minus 60 molar, then those of you who remember Avogadro's constant, 6 times 10 to the minus 23, will realize that there isn't any glutamate in either of these columns. So either homeopathy is alive and well at the NIH, in which case, of course, the 10 to the minus 120 molar should be the more powerful treatment, being the more dilute. Or these findings don't fit either with the precepts of conventional biomedicine or with homeopathy. So there's something wrong with the way that we do these experiments also. So some years ago, we looked at drugs that had been developed for stroke. My day job is I'm a neurologist, I'm a stroke neurologist, and I want treatments that I can give to patients quickly. So are there drug treatments that we know are safe in that population that we could give to patients very early before the diagnosis was completely secure with little risk of doing them harm? And we identified over 1,000 compounds tested in the laboratory for stroke, of which 600 improved outcome in that animal model I just told you about, of which remarkably almost 400 improved outcome in that animal model of stroke, like low-dose glutamate, like Arnica, of which 97 had been tested in human clinical trial, of which one clot-busting treatment with TPA actually improves outcome in that clinical trial. So, and TPA tested in human stroke, not because it works in animal stroke, although it does, and we've got a systematic review that shows it does, but because it works in the cognate human condition myocardial infarction, where the blood supply to a critical organ bed is occluded by thrombus. And we've got 3 treatments that work in humans that have never been near an animal, so stroke unit care, aspirin, decompressive hemicraniectomy. So depending on how generous you're feeling, the attrition rate here is either 1 in 1,000 or 0 in 1,000, but either way, it's not very good despite all of the investment over the years in developing stroke treatments. So here's the problem we have with translational research. We take findings from what's supposed to be a mouse in the cartoon, and we translate it across some species barrier or translational barrier to apply it to a clinical trial in human, here of the beneficial effects of red wine. And I like to think also about the possibility that we might choose to do what I call cislational research, that we could say, well, actually, we don't have enough information just now to move into humans, so we're going to continue to do more research in animals until we get to that point. And so if the sum of existing knowledge is less than some threshold, then we should do more cislational research. And if the sum of that knowledge is greater than a threshold, we should translate. But the problem is, we've got very little way of knowing what that threshold is and where it is, because we don't understand the research that we do well enough, and we don't have ways of aggregating together evidence from different studies in a form that's meaningful against this threshold. But the key question for research investment is, what's the next piece of what's the best marginal utility of the next piece of research? How do I maximize the value of my research investment? So as you know from Johnny Unidis, most published research is false. So a lot of the data that we're planning our next research on the basis of isn't true. So if we take a power of 20% and an alpha of 5% and assume that the proportion of studies that are truly positive is about 10%, the chances that a statistically significant finding is actually true is only 31%, which goes a long way to explaining some of the replication crisis. So this is in a cartoon from a commentary I did with Jeff Mogul. So this is thinking of a stroke example where you've got 250 compounds, 50 actually improve outcome, 200 are inert. We are powered at 20%, and we know that most studies in the neurosciences are powered at 20%. So we detect 10 out of these 50 drugs that do work. We take an alpha of 0.05, so of these 200 compounds that don't work, we identify correctly 190 as being negative, We falsely identify 10 false positives, and that means that actually we get 20 positive studies, and in those 20 positive studies, 10 are truly positive and 10 are falsely positive, so the positive predictive value is 50%, not 95%. And that's interesting because to a certain extent, some of that information is already taken into account in the knowledge of investigators. So Anna Dreber from Stockholm, who I understand from Olavo is your speaker next month, is an economist by training and has done this fascinating and really important work. And she shared some of the underlying data in this with me. So for, I think, 42 studies that were included in the psychology replication project, they ran a prediction market where a large number of psychologists of varying levels of seniority were given money to invest in a research finding. And if that research finding that was going to be tested in the replication study, and if that research finding was confirmed, then they got $1. And if the research finding was not confirmed, they got nothing. And so they just let the prices of these experimental findings float as people invested in them or disinvested in them according to their beliefs about whether it was true or not. And she was interested predominantly in going from the originator study P0 here to the before the originator study to the study which was included, which was the finding to be included in the replication study. So this is where the replication study started, and this is where it finished. And what she was interested in is using their prior beliefs here and knowing the powers of the initial studies to work back to say what was the prior belief before they started. So because you know the power and you know what the performance was in the p-value, you can say, well, before the first study was done, the prior belief in the community was. But and then they said, and isn't it interesting because some of these studies replicate and others don't. So this is the posterior after the replication study, the posterior beliefs. And here she kind of gave me the individual data points for all of these. Now, what I'm most interested in here is the transition from before the replication study at P1 to after the replication study at P2. So for each study, P1 is the prior for the replication effort derived from the market that she ran. P0 is the calculated original prior before any experiments were done, and P2 is the posterior. Now that P1 relates firstly to the strength of the original evidence, and secondly to expert critical appraisal of that originator evidence. And for each study, we also know the power of the replication study. So we can predict the probability of successful replication, P1 times the power. And averaging across those 41 studies, we would expect a probability of replication of only 0.53 based on the community prior and the power. And actually the non-replication rate, the probability of replication 0.353, probability of non-replication 0.47. So we would have expected, based on prior beliefs in the community at the time, we would have expected 19 of those 41 studies not to replicate. And we observed 25 of the 41 not replicating. So actually of that replication crisis, replication failure in psychology, 3/4 of it has already been taken into account in the, if you like, the hive mind of the community about what's true and what's not true. And I think that's an important thing to bear in mind, and it might be possible to build in something like this, Olavo, into your own prediction markets. So most individual studies and most research summaries are at risk of bias, and that's because there's designs which introduce risk of bias, or because you don't actually have all of the research items to summarize because of publication bias or because of selective outcome reporting bias. So this is our work for NXY059 in animal models of focal cerebral ischemia developed by AstraZeneca for the treatment of stroke. And after 5,500 patients in clinical trial, there was no effect at all. Their share price fell by $9.6 billion in 48 hours, and it took them 7 years to recover. And with the benefit of hindsight, we went back to look at the animal data, and you can see that overall, this gray bar is the 95% confidence limits of the overall estimate of the treatment effect. overall NXY059 is great in animal models, improving outcome by 44%. But when you look at those studies that reported randomization, that reported blinding, that reported the blinded assessment of outcome, then their efficacy was substantially and significantly smaller. And of course, that was 2008. We did that study. This has been known for years and years now. And again, I apologize if you already know the answer here. Robert Rosenthal, great guy, described the file drawer problem, described the Pygmalion effect in education, and described this effect. He taught a course in animal psychology to graduate students, and at the end of the semester, they had to do an experiment. And the experiment involved comparing cognitive performance in two cohorts of rats, one of which had been selected for many generations in Berkeley, at that time a great center for cognition research, to perform well in cognitive tests, but they were naive to this particular test. And the other were their slightly dull, inbred country cousins selected to perform poorly. The task was pretty straightforward. It's a T-maze. One arm here is lit, the other isn't. This unlit arm is always reinforced with a food reward, and which arm is which is flipped according to a predetermined random schedule. So the rats, rather like our junior researchers, are being trained to turn to the dark side. And this is what they found. The mazed bright animals started off performing better than the mazed dull animals. Both cohorts of animals improved over the 5 days of the experiment, but that improvement was much more substantial in the mazed bright animals, as you would expect. And when he asked the students what they thought of these animals were bright-eyed with lovely wet noses, the sort of animal you'd like to take home to meet your parents, whereas these were mazed dull animals were surly and insolent. And in fact, 3 of the students were bitten by the mazed dull animals. And in fact, the experiment that he'd done wasn't on the animals at all, because they'd been drawn at random from the same cages in the same animal house on the first day of the experiment. The only difference between them in their cognitive performance and in the minds of the, and presumably also in their biting behavior, the only difference in performance must have been in the minds of the investigators and how that transferred to the students, to the animals under test. So you would expect, when we started doing all of this, we were told that everyone knew that researchers in neuroscience were stupid, researchers in stroke were stupid. That's why we didn't have any good treatments. And so we started to look at other neurological diseases to see what the impact of risks of bias were there. And so I seem to have missed my slides. And what you can see is that in stroke, in Alzheimer's disease, in multiple sclerosis, in Parkinson's disease also, then studies which didn't report blinding give substantially higher estimates of treatment effects. So the green arrows show the studies at low risk of bias. So Alzheimer's disease, that's Morris Water maze. So it's video capture of behavior, but if the investigator knows which group the animal belongs to, somehow it transmits their behavior. In Parkinson's disease, that's the 6-hydroxydopamine unilateral striatal lesioning model. When I was young, I was told it was a bioassay. Well, it's only a bioassay if you know the treatment group to which the animals belong, and if you're blinded, the treatment effects are much more nuanced and much more subtle, and probably below the limits of detection. So what I thought I would do is I would stop there if there's any questions, and then after that we can move on to the next set.
I was muted, sorry. Yeah, so looking for questions. I just sent you can send questions, and I sent it to the wrong, so Juliana just resent you. So I'll start off while people think of their questions, but you can either make questions via chat, you can write them and we can transmit them to Malcolm. You can also, if you're in Zoom, you can also raise your hand, then we can put you, Juliana can put you live if you want to speak. So I'll start off with a question though. I mean, you talked a little bit about sampling and like non-random, like how sampling is usually not random, and you did make your claim that like we should probably expect that most research, most fields of research have reproducibility problems. in terms of in the few that we have tested, we do find them. So on the other hand, it's terribly hard to replicate. I mean, it's not easy to put up these replication initiatives. I mean, it's costly and it takes a lot of people and effort to actually replicate studies. So where would you start and how would you sample? Like how do you define which fields are a priority in terms of replicating, and what's the best kind of sampling? Because I mean, it's never going to be random, right? Even if you have a big group like we have, you still are very, very limited by expertise. So what would you? I mean, we're taking our best guess to do it in Brazil. But like, what would be your best approach in terms of like how do we select experiments for application?
You mean if I ruled the world? Yes.
Oh no, if you had a lot of money to do a replication initiative.
So I'll come to this a little bit later in terms of the finances, but the costs of poor research are huge. So it's been estimated that 85% of research is wasted. That is several hundreds of billions of dollars a year. So I don't think we should be constrained by budget. What I would do is I would sample thousands of scientists and get them to nominate their 10 most important findings in their field. Or I would go to the citation banks and take the most highly cited publications, or I might even sample in highly cited, medium cited, lowly cited publications. So there would be a way of getting a long list of findings. I then look at those findings and extract from them relevant information. So the relevant information would be the field of research, the techniques used, the number of authors, the decade of publication, the country of origin, the journal of publication. And then I would do it like a clinical trial where I would sample from that in a way with minimization to get representation of all of those different kinds of research with sufficient statistical power to be able to tell a difference. And then I would fund replication studies for all of them. So every lab in the world would have to stop doing what they're doing for 6 months to make their contribution to this global replication effort. And then I would work, and at the same time, I would run alongside that a prediction market, and I would work out who was best at predicting what findings. So I'd put the people, and then what I'd do is I'd say, well, we know these findings, I've got a high chance of being true because they replicated these, I've got a lower chance of being true. Do we need to do multi-center studies to try and get the real answer for this particular question? And then the people who scored best in the prediction market, I would put on all the funding panels so they can make the decisions about what research to invest and what research and not to invest in. So that's what I would do if I had, this is a tractable problem. Tracting it is very, very expensive, but that's how you would do it. And like anything, if I'm trying to get a large study funded and all the funders are going, yeah, we're not going to give you that much money for this, then what I would do is I would do a pilot study which showed that, that showed proof of confidence that we could do it, that it was possible, that it was feasible, whatever. And then I would put together the results of all the different little pilot studies and say, here's a bigger study. And then I would do, so I would gradually work my way towards that objective. And I think that's what you're doing just now. I think that's what the other replication networks are doing. I'm sitting on a funding panel in Germany in a couple of weeks' time where they're funding replication studies. And they're also using a registered funding model in that. So for sure, their replication studies are going to get published because that's going to be built in with it. So, we're not going to get there tomorrow, but it's we'll get there slowly. We'll get there slowly. Yeah. Do you want me to read some of these questions?
Yeah, so we have a couple of questions via text. One is from Selene, who does not have a microphone, but who was asking about effect sizes and sample sizes. I mean, should we keep doing small studies? Should we gradually move to?
Yeah, we should definitely stop doing them because.
I think meta-analysis helps.
Well, meta-analysis helps a little bit, but if you have small studies and publication bias, and I can't remember if I come on to, I think I took those slides out, but essentially small studies will overstate biological effects. Let me just see in the background here if I do get to this. Yeah, I'll come to this. In fact, let me tell you this now, and we can skip over it when we get to it. So this is, can you see my slides in the background now? Yeah. Olavo, you can see my slide? Yeah. So this is from a systematic review of experimental allergic encephalomyelitis. And you see that in this is study size, small studies give larger estimates of effect. And if you do a simulation and assume a complete publication bias when anything more with P of more than 0.05 is unpublished, and do a meta-analysis of the studies that are published, then you get this picture here. And so small studies will almost always overestimate treatment effects. Even when you're powered at 86%, you're still getting an overstatement of the efficacy. So you've got two sets of studies. These studies are underpowered. These studies are adequately powered. The overall estimate in meta-analysis is just the same. But if that study is missing from the literature because it was never published, then we estimate this effect size here, the green effect size in the other, and we overstate efficacy. So there is, to my mind, no excuse for doing small studies. And if that means joining together with other groups to answer the same question, then that's helpful. So Kate Button is running a program now for undergraduate projects in psychology, where these are undergraduates, they don't have enough time to do everything, but she's yoked together 4 different institutions, all of whom have a graduate course in psychology, master's in psychology. So they all work on the same project and get a publication out of it. So there are ways of increasing your numbers.
Interesting. Yeah, so this is Martin Spitaler here. If notes are as likely to be right or wrong, how come life sciences have moved at such a fast pace, at least in basic research? I do think that's an interesting question.
So I think that we've moved on reasonably quickly. But if you look at the outputs, if you were to read your equivalent of our Sun newspaper or Daily Mail, you would think that scientists were discovering cures for cancer every day. The early work from the RAND Corporation and from Ioannidis suggests that it takes about 20 years from a target being identified to that drug getting into the clinic. So that doesn't seem to be very fast. And the number of new medical entities, unique new medical entities coming for regulatory approval has fallen dramatically. Now, what I think has happened, by way of explanation, and in contrast, propranolol, the blood pressure drug, went from chemical synthesis to chemists' shelves in 3 years. Now that's because propranolol has got huge biological effects. So I think there's a limited number of compounds which can interact with us biologically. I think the compounds which have got large effects, which interact powerfully with our bodies, have largely already been discovered by nature, by evolution, and if not by evolution, by early chemists. And so the effect sizes which had to be observed were large. And in the context of very large effect sizes, the reproducibility of those findings will increase. Statistically, that's what will happen. But as the effect sizes available to us to try and make druggable fall, then the statistics become more challenging and come into play more. And we've done a little cheeky regression of, we've got something like 20 or 30,000 outcomes from meta-analysis in our database now. And if we regress them against time, we predict that by 2060, there will be no efficacy on average in biomedical research. It will, because the efficacy is just falling over time. So
is that in humans or mice or rats?
In rats and mice, in laboratory animals. The meta-analytic estimate of efficacy across all disease models falls by about 1% per year.
That's a great
and that's because the interventions which we're choosing to test are less powerful. because the more powerful ones have already been discovered. And you know, it's difficult to imagine that there's a chemical compound with the effect on blood pressure of the same magnitude as propranolol that someone hasn't discovered yet, or that millions of years of evolution haven't discovered yet. I just think that's implausible. So I think that the reason is that, first of all, it was fast before, not quite so fast now. Of course, science precedes breakthrough at a time. So CRISPR-Cas has just opened up a whole new avenue of potential approaches to biomedical research in the way that antisense did, in the way that our understanding of apoptosis did, zinc fingers, talons, whatever. And so there's a big boost of new knowledge when these come through, and that's understandable. But how much of that actually makes it through quickly to new treatments? Well, I don't know. I don't know. When I was doing my PhD actually in the lab 30 years ago-ish, 25, 30 years ago, we were talking about antisense and how antisense was going to give us great new treatments for diseases. And last month in our medicines regulators, we saw the first time I'd seen an antisense-based drug for therapy for a genetic disease. So that was 25 years from thought to. So I don't think it's that fast. I think it could be faster.
Do we have any more questions at the moment? If not, I think we can move on to the second half of the talk, and then we can have more questions at the end. So
okay,
go ahead.
Yeah. So so I'm interested in how we can make things better. I don't think there's any point in saying how awful, how terrible things are. We should just give up. We've got a thing in the UK. You've probably got something similar in Brazil. We call it the Research Assessment Exercise, although they've changed its name now to the Research Excellence Framework. And in 2008, they have them every 7 years. In 2008, they concluded that we were really pretty good in the UK. We were outstanding, really, and they were particularly impressed by the strengths in neurosciences. Now, that kind of was a bit different from what we had thought from our work. So we set out to identify the leading UK institutions. And they've got different units of assessment. So we took the institutions which performed best across the 6 units of assessment where in vivo research was focused, was concentrated. And we found 1,173 publications using non-human studies published in 2009 or 2010. This was done in 2008 from 5 leading UK universities. I'm proud to say that my university was considered to be a leading UK university. And then we ran these against the features that were identified by Story Landis in her reproducibility transparency paper as being important to allow a scientist to do due diligence on whether a research finding was true or not. Randomization, blinding, reporting inclusions and exclusions, and the power calculation. Now, these institutions are color-coded to protect my future career. Lovely Scottish color here. Randomization reported by less than 15%, blinding by 18%, inclusion and exclusions by 10%, sample size calculation by less than 2%. So this is shockingly poor for leading institutions in one of the leading research, we like to think, one of the leading research countries in the world. Only 1 out of 1,173 did all 4 of those things, and actually 68% did not one of those things. So there's a problem. We've done this. Same slide. Then the other issue isn't just about the risks of bias in the individual studies. It's whether the individual studies are described well enough to allow you to try and replicate it. So this is Kieran Egan's PhD work on testing cognition in mouse models of Alzheimer's disease, drug or not, in the Morris-Water maze. And this is the size of the Morris-Water maze pool used, and it varies from 85 centimeters to 2 meters. This is the size a mouse pool should be. This is a rat pool. But in 20% of the cases, the publication doesn't tell you the size of the pool. This is the water temperature that was used. This is the main verse of stimulus to get the get the animal to try and find the platform. This is 16 degrees. This is Scotland in the height of summer. This is probably Copo de Cabana any time of year, but over 40%. No, not that warm. for 40% of the time, they don't tell you the water temperature. And this is the training, the number of acquisition trials per day, and the number of days training, and the modal responses that they tell you neither of these things. So I would hold that it's impossible to seek to replicate the Alzheimer's Morris water maze literature because it isn't described in sufficient detail to make that a realistic prospect. And that's a problem because it may be, this is Bernard Vocal's work with Hanno Verbal, it may be that both studies may be true. So imagine that you've got some biological response here, could be the infarct volume thing that I talked about earlier on, and you've got some nuisance variable, could be the noise in the animal house or the perfume of the investigator or something like that. And I do my experiment here and get this result, and someone else does their experiment here and gets this result, and we go, oh, this is terrible, this is a replication crisis, when in fact, we've just sampled the same biology, but at different levels of this reaction norm. And there's a great example of this in the literature, actually. So there was a story that many years ago, a group reported that a mammalian target of rapamycin inhibitor could extend lifespan in C. elegans. And there was lots of excitement because this was the first time in any animal model that a drug had been shown to extend lifespan. So immediately two groups tried to replicate the finding and they couldn't replicate the finding, and everyone said, same old, failed replication. We know this happens, walk away. And then several years later, another group showed that a similar drug, another mTOR inhibitor, extended lifespan in mice. And some really bright spark at the US National Institutes of Aging remembered the worm story and said maybe they had something. So they put the worm gangs back together, the three original labs, which had the originator lab and the two replicating labs, and they funded them, all were by that time based in the States, all the PIs, to try and see if they could get rid of this variation between studies, see if they could understand what happened. So they managed to identify sources of variation even when they tried to get everything sorted out. So these worms live on a feeder layer of dead bacteria. So they all use the same bacteria, all from the same stock. Sometimes they tried to see if a worm was dead by, some touched it by a metal pin, some touched it by a brush. They standardized all, even when they standardized all of those things, there were sources of variation which were genetic, fine, in the developmental rate and in fertility. There was between lab variability, there was some variability within labs, and some variability that was individual. And this is in C. briggsii, not in C. elegans, but if I just draw your attention to two of these strains, AF16 and JU726, in three different labs. What you see is that in this lab, AF16 has a monotonic death response. These are Kaplan-Meier survival curves. All the worms are dying at the same rate. In this lab, for the JU726, all the worms are dying at the same rate. But look here, the JU726s in buck, there's a bimodal death response. Some are living much longer than the others. And in the AF16s in Rutgers, again, you've got a bi or trimodal death response. So in spite of trying to control for everything they could, there are some differences between these labs which are driving these really quite profound. This is a 40% difference in median survival, really quite profound differences in lifespan, which we could have known about 20 years ago if we hadn't been prepared to accept failed replication as an explanation for everything that we can't see. And in my view, an awful lot of the failed replication will be because of this variation and underlying latent independent variables. And that's why multi-center studies are so important, because you, by going to different labs and different groups, you sample some of those latent variables which are in play in replication studies, which aren't in play in the originator studies unless you do them as multi-center originator studies. So what we should do, we should increase the probability that published research is true, establish a framework to select efficiently which research findings we should attempt to replicate. I talked a bit about that earlier on, and develop strategies to evaluate the robustness of key research findings, multi-center studies, those are. So if we change our setup here, so we're powered at 80%, not 20%, and if we take a P threshold of 0.01, not 0.05, we'll need many more animals, about 7 times as many animals, but our positive predictive value now is 95%, even if our prior is only 20%. So this study now does what we thought the previous study should do. And researchers are very different in their qualities. So you can imagine a distribution of researcher quality, and some people are down here, and a very few number of people are up here, and most of us are somewhere here, and we aspire to get better. But our institutions, and so the things that define this are falsification, fabrication, and plagiarism, hypothesizing after results are known, doing experiments at risk of bias when you know they're at risk of bias, and then moving on to better things like open science practices. And I should say, I've tweeted to your Twitter hashtag a link to these slides, which are on the open science framework and are CC by NC, so you can all use them, take them apart, however you want to do. And then on to, pre-registration and registered reports and all of that, which are, very high quality research practices. But our problem is that our institutions and our efforts rest largely on saying, who are these people? Let's hunt them down and kill them, or at least fire them and make sure they never work in science again. And that doesn't really work because there's very few of them. And it means that every time my institutional research improvement officer comes round, it means that I think that they either think I'm at it or I'm stupid. And instead, I think we need to shift the way that we think about research improvement to say, if this is the distribution now, let's get everyone just to be a little bit better. And if everyone gets just a little bit better, then overall, the value of the research that is done will increase substantially. And how might you do that? Well, you need to measure the outputs of research. And we've been developing tools, and Celine and others have been involved in developing the tools for this that would allow us to measure performance against target automatically by annotating by risk of bias and by content, test interventions to improve research quality, and then in terms of translation, provide real-time summaries of existing knowledge. I was speaking to a student who was coming for a job yesterday. She'd done a systematic review. From her search to the systematic review being published was 2 years. And systematic reviews are the most complete way of summarizing existing research. So if we're going to make decisions based on the best available evidence, we need to have systematic reviews which are done much more quickly than 2 years. Providing evidence to guide future research, this cislation translation thing that I was talking about. So here's a corpus of publications in stroke, analyzed with machine learning for the reporting of blinding. These are journals which have reported more than 5 publications during the duration of the study. And you can see some, they're all blinded. Some are very poor. This is the average here. This is the overall performance, about 70% in the focal ischemia literature now. And here's +1 in green. So if anyone tells you +1 is rubbish, well, it may be rubbish, but it's less rubbish than probably 60% of the other publications in this space. So we had a statement of good laboratory practice for stroke modeling published in the journal Stroke a couple of years ago, 2009. Mark Fisher, the second author, was the editor of Stroke at the time, and he implemented a new editorial policy that reviewers had to tick a box for whether or not this was randomized. Jens Minnerup and colleagues have gone back to look at the performance in Stroke since that change. We weren't involved in this evaluation. And you can see reporting of randomization increasing by about 30%, validation concealment by about 20%, of blinding again by about 20 or 30%. So that's improved things at that journal. How does that, is this just because everyone's getting better? Well, these are the 5 American Heart Association journals: Circulation, Circulation Research, Hypertension, Stroke, ATVB. The adjusted odds ratio adjusted for cardiovascular disease studied and the animal model used. And you can see that there's an uptick in stroke across all of these, not seen at any of the other journals. So it's what Mark did specifically at that journal, getting the editorial editors to, the peer reviewers to complete the checklist, I think is what helped. We did a study, again, which some of you were involved with Nature. So Nature changed their guidance for life sciences publication in 2013. And we worked with them to identify 400 papers across Nature journals published after that date, to match them for subject area to Nature journals published before that date, and to use Google related citations to match them to another 2 lots of 400 journals. Sorry, 200, 200, another lot of 200 plus 200 in other in journals from other, in manuscripts from other publishers published before and after in the same topic area. And what I'm going to show you here, the black is that they said we randomized, and the grey and the hatched bit is where they mentioned randomization, but only to say we didn't randomize. So you can see that Nature had a big increase in reporting, largely but not exclusively, of people admitting that they didn't randomize. But that allows me to analyze the paper, allows me to give credence or not to the findings. In other journals, no change. This is let alone what the nature of the study was. And then we used crowdsourced outcome reviewers to assess whether the study met the arrived checklist. And we randomized 1,600 papers. About 660 were accepted for publication in PLOS One. Of these, 13 of the control group had sent in a checklist when they'd been asked nicely. 300 of the intervention group had sent in a checklist when it had been required. So still, if you're stroppy and phone up the editorial office, they'll let you through without a checklist. And the number of papers that were compliant with the arrived checklist was 0 in both groups. So if you isolate down getting people to complete a checklist, which is often promoted as a way of fixing this problem, that in and of itself isn't sufficient to change behavior. So we need to be smarter about how we try and change behavior. So every institution which conducts research should have a formal research improvement strategy. Funders should consider the high returns on replication studies because of the high chance of revising existing knowledge. You know, I showed you for the replication studies that they confirmed the finding 30 to 60% of the time, which means they refuted the finding 30 to 60% of the time, given that they were of adequate statistical power, so that the chance of a replication study advancing knowledge, even if it's advancing knowledge by leading to a knowledge claim being withdrawn, is better than funding primary research, more likely to be fruitful. And pivotal research findings should be challenged in prospective multi-center replication studies. 300 billion a year globally, 50 billion in Europe. Even if waste only so even if that waste is only 50%, and even if measures to increase research value only reduced that 50% to 49%, we'd free up $3 billion a year for biomedical research. So investing 1% of research expenditures in improvement activity would go a long way to making things better. Thank you very much. Sorry for going on so long. Happy to take questions.
Okay, so we're open for questions again. Again, once more, you can raise your hand or you can ask via text. Our Facebook Live, I think, froze for a while. It's back now? No. Yeah, so yeah, we're back. We're back. Sorry for the pause if you're on Facebook. I do have questions again. I might as well start. I actually have a couple. One on the checklist on the checklist matter. Why do you think stroke was so different from PLOS One? And do you think the checklist problem in the end is just where it's happening? I mean, again, journals are asking once you've already done the studies. I mean, can you figure out like a better way of actually having a checklist at the beginning of the study in some way?
So yeah, so to take the second bit first, some people have argued that you have a iterative approach, that when I get my checklist for my study now, it's too late, but when I plan my next study, I think, oh, I'll take that learning forward to my next study. For the minimum standards framework checklist, we're calling the MDAR checklist, materials data analysis. Forgot what the R is. That's terrible. Reporting. We're going to be piloting that with authors as a tool for writing their manuscripts, and we're going to have supporting materials around that to try and help. When Stroke had the checklist on their editorial website, when the peer reviewers put in their editorial comments, you know, is your recommendation accept, reject, whatever, do you have any conflicts of interest? They had a box that said, does the study report randomization, yes or no? And if you didn't complete that box, you couldn't submit your peer review. So it forced the peer reviewers to pay attention to whether or not studies met these essential things. And Mark said, he said, I'm just not going to publish stuff, or you know, I'm going to do my best not to publish stuff that's not been randomized or blinded. So I'm going to send it back. And if you're not randomized or blinded, you need to say that absolutely, in your paper. And so it wasn't so much that, you know, it was a checklist that the journal was supposed to follow, or it was a key part of the peer review process that you couldn't get by unless you did it. And so that would work for some journals to do that. The trouble is that when you try and operationalize these checklists, not just randomization, blinding, and the like, when you try and operationalize these checklists, they come to hundreds of components. So you would have a peer reviewer, and those of you who did the Icarus study or the Nature study, who had to review the papers against the operationalized checklist, will know how troublesome it is. It takes ages and ages and ages. It takes about 2 hours per paper, and to get peer reviewers to do that, you would just have people not doing peer reviews anymore. So our advice, I think, is along the lines of trying to, instead of saying to people you need to do everything at once, is trying to say what are the most important things to fix first, and let's try and fix those. And then once they become embedded in the habits of the community, we can then move on to the next thing. So I think it likely, although not definite, that the new version of the Arrive checklist, which is coming soon, is going to have 20 items, but it's going to have a top 10. And so it's going to say to journals, you know, if our recommendation is you try and do all of the stuff, of course, but if you need to focus on something, focus on these. The MDAR checklist is going to have things that are considered to be essential, things that are considered to be less than essential, with the idea that journals might start their journey where, you know, what I'd really like to see is have people having self-assessment tools. So you could say, I've got a journal, or I've got an institution, or I've got a funder, I'm interested in this improvement journey. I need to assess what my current performance is. And then depending on my current performance, I'm going to pick a tool that's going to try and make me a little bit better.
So I have a couple of questions here. One is, Selene asks if multi-center studies will reduce the number of animals in research, or can they help in that? And second question is from Juliana Camacho here from our institute, which is also about multi-center experiments, but a separate question, like if we are to always match with other groups to increase the number of experiments, size, and reproducibility, we might lose research that's original, because, like, no, it's going to be hard to find a lot of people who find this is interesting. So won't you lose disruptiveness in research in some way?
So a couple of things. So to Charlene, yes. So Hanno Verbel's group did a simulation of this. I think Bernard Vocal was the first author.
Yep.
It's in PLOS Biology. Emily Senna's on the paper, where they simulated using animals in a single center study or using the same number of animals in a multi-center study. The precision of the estimate in the multi-center study was less precise, but that finding was more likely to be replicated in future studies. So it was a better estimate of what they were getting. So if you'd get 36 animals in a study, your study is more powerful if you do 3 studies of 12 animals across 3 labs than if you do 36 animals in 1 lab. So I think there's a lot of motivation to try and do that. And that ties in nicely, thanks. That ties in nicely to the second point, which is, how do I do this? Because I'm doing stuff which is pretty, you know, I want to be doing research that nobody else is doing. I want to be doing research that's cutting edge. So how can I do this at the same time? So I think there's a phased model which works, or I think might work, which is to say, all the stuff you do in your own lab, that's, in fact, even though you might not quite think it, because you're not taking into account the latent variables between labs and all of that, it's largely exploratory. You probably don't have the size to get the statistical power to do a proper hypothesis testing study in your lab. So what what Jeff Mogul and I suggest in that paper is that you have this phase of work which is almost explicitly exploratory, where you try and develop the information and the case to go into a hypothesis testing confirmatory experiment. And that will then allow you, because if it's not hypothesis testing, you don't need to do statistics. So you shouldn't really need error bars and all of that. The only person you're trying to convince is yourself. And then once you think the data are in, I'd like to see people similar to labs like that joined up in consortia who agreed to test each other's hypotheses alongside their own work. So people that were using similar disease models would, so you would do a bit of work for them testing their hypotheses, and they would do a bit of work for you testing your hypotheses. And you could have it if you pre-register your study on the Open Science Framework. If you've got a registered report, then you can demonstrate that you were first to have that idea. So you can demonstrate primacy in getting to this explanation of how the world works, which I understand is often a problem. So how do I involve enough people to deliver the work while not involving so many people that someone else scoops me? And so that's the way that we thought in Multipart that it might work. And I know that the neuropathic pain community have been thinking similarly, although their community is largely North American, so they're more worried about how much money will change hands and in which direction than are us Europeans. But just for the sake of argument, say I'm doing an experiment with 16 animals using my model, whatever. Can I add 4 animals using the same model that's an experiment for a labo? And if he does that, he's got his 16 animals, and then he's got 4 sets of 4 animals from 4 other labs. He's up to 32, and you could call that hypothesis testing multi-center experiment. And then if that was right, you could then go into a proper confirmatory phase 3 multi-center animal study with funding.
How do you see publication working in that way? I mean, I like the model, but I mean, would you have like 2 publication tiers? You publish exploratory work, and then you publish confirmatory work, and this is like clearly marked, or do you just publish at the confirmatory phase?
So I think this may be a conversation we don't need to have in 10 years' time, because I think the publication model is going to change dramatically, and that Plan S is going to help that along, but also all of this stuff as well. My preferred publication model just now is bioRxiv. That I've got something that I think is ready to publish, and I put it up on bioRxiv. Now I've got many papers which have been substantially improved by peer review. So what I would like to see is studied data, pre-registrations, whatever, in a repository, Open Science Framework or something like that, with bits made public by the investigators as they go along. Then some kind of bioRxiv type paper which articulates the hypothesis, and that might be part of a registered report or a protocol for the study. And then the study itself, I think, should be published in bioRxiv. And instead of us chasing journals, what should happen is that journals should scan bioRxiv and say, this is something that we would want to publish. And they should approach me and say, we'd like to take your paper. We'd like to give you the opportunity to improve it, because we're going to send it to some experts and they're going to comment on it and make suggestions for improvement. And if we go through all of that, we'll then promote this, disseminate this through our channels, be they electronic or print or whatever, and you'll give us some money for the contribution that we've made to the dissemination of your research ideas. Now, the other issue that's wrapped up in this is credit for authorship, particularly for young researchers, for junior researchers. How do they get their career to move on if they're part of a science team with 10 or 15 people in it, where everyone's made a contribution? How can something be theirs? Well, firstly, that relies on a misconception that anything in science these days ever belongs to one person. I mean, there's bits of projects that we give one person to take the lead on, but it's always, in all my time in science, it's been a team effort. And the people who sit on their own in a corner and don't speak to anyone, they're the ones that you don't trust as much. And in the UK, and I think elsewhere, there is more recognition of this team science thing. DORA, San Francisco DORA statement that my institution, I think, is about to sign up to, has good guidelines for how you use metrics like publication for assessing staff for promotion. And it says you shouldn't use publication, citation, bibliographic and matrices. Having said that, I gave a talk yesterday where one of the postdocs came to me and he said, we'd got an internal competition in the university last year to allow you to apply for external funding, startup funding from a national organization. And a whole load of us weren't selected because we didn't have papers in Nature and Science and Cell. So I get that it's a problem. I get that it's not easy, but all of us who are in positions of some authority within institutions, every time we hear someone say, but it's a Nature paper, we should say that means bullshit, that means nothing. And every time someone says, well, they've only got some papers in bioRxiv, you say, well, have you read them? What's your assessment of the quality of that research? And don't just look at the cover of where it is. Read the research and judge the quality by reading it, not by some kind of surrogate measure of quality, which have been around for so long that they've been gamed absolutely to death. And so the people in the higher echelons of our research infrastructures are people who have learned, I mean, it's not deliberate, but they've just become conditioned over 20 or 30 years in the trade. They've been conditioned to know what works under the status quo. And what works is doing rather small studies, rather underpowered studies, testing a whole load of really quite extreme hypotheses. One or two of them will pop out, and then navigating your way through doing the confirmatory figures, 3, 4, and 5 requested by the journal in a way that gets you an answer that's publishable. Now, those aren't the skills that we should be engendering in our next generation of researchers, or indeed in our next generation of research leaders. But it's not going to change unless we take up arms in this revolution to improve the way that biomedicine is done. Every year in Scotland, a tiny country, 5 million people, every year in Scotland, 400 people have motor neuron disease, about 100 of them die. And when we eventually get a treatment for motor neuron disease, ALS, I'm sure we will, say that treatment improves quality of life by 1 quality adjusted life year. And I could give it to all of my 100 people in Scotland. And the 400 people in Scotland at any one time with MND, 100 incident cases a year, that means I'm doing 100 quality adjusted life years in Scotland, just like that, from this new drug. That drug will have been published in a paper. That paper will have been delayed in its dissemination while waiting for one of the major neurology conferences to be presented at. There'll be an embargo on releasing the findings of that paper. That clinical trial will have been based on some earlier studies, probably which also had an embargo, probably which also took at least 6 months or a year to get published. They'll in turn have been based on animal work, maybe 2 or 3 animal studies in sequence, each of which will have been delayed for 6 or more months getting published as it was submitted, peer review, revise, reject, resubmit somewhere else. They'll have been based on in vitro research, which will have been revised, rejected. So in that long chain of publications, that long chain of research that's got us to this treatment for motor neuron disease, which Ioannidis says can be up to 20 years long, 5 or 10 years of that is the delays which are engendered by our publication system. And 5 or 10 years of ALS, for me, is 500 to 1,000 quality adjusted life years in Scotland every year for patients with one disease, which is a very uncommon disease. And if you map that out across all of our diseases across the whole world, the costs in human misery and suffering of our current publication model are huge, and I think are unethical. And then I'll tell you what I really think.
Yeah, I fully agree. We do a lot of that. We do have more questions still. Otherwise, I think this is a great. Oh yeah, we do have one more question. So, why do you say the faking data is low in clinical studies? It's really more difficult to maintain for long a fake finding statement. So, how do we improve the possibility to distinguish in large scale the fake findings from the bio? I mean, it's mostly
so I think so in both clinical medicine and
tracking fraud and false data or not.
Yeah, so I think in clinical medicine and in psychology research, and to a lesser extent, I think in biomedicine, there have been 1 or 2 famous cases of people who just made up the data. We had a case in the UK where someone who was working in a CRO actually got sent to prison for faking an assay. So there was an assay using serial dilutions, and if you didn't put in a blank between each serial dilution, you got carryover and it screwed the assay, and he suppressed these whales. Anyway, he gets sent to prison for 3 months. But I mean, maybe I'm just an optimist. I don't see scientists really who deliberately invent their data. I do see scientists who will do things that they think are acceptable because they've never been taught that they're not acceptable. So they'll get a distribution of answers and they'll say, well, you're allowed to exclude everything that's more than 3 standard deviations away from the mean. So they'll smudge out that data point, and then they'll do it again, and then another outlier will now be outside 3 standard deviations, and they'll smudge out that data point, and they'll smudge out. So I'm sure that happens, but that's through ignorance rather than malfeasance, you might say. I'm sure that people do one statistical test and they don't get the right p-value, so they do another statistical test, and they keep doing it, and then they log transform the data and arc sign it and do whatever until they get the answer they want. I'm sure that people measure 3 outcomes and only report, and if only one suits the hypothesis, only report the one. We know about image fabrication and falsification. There's a bit of stuff around falsification that was Anita, forgotten her second name, I can tell you later, who's interested in RRID, so research identifiers, robust or persisting research identifiers for research materials. If you combine that with, for instance, the oligo sequences that are reported, so are the antibodies used the right antibodies? Often not. And if people find that the antibody gives them the pictures they want, they'll stick with that antibody. There's a consortium out of Monash who are interested in whether the cited oligo sequences in papers are correct. And many of the cited oligo sequences are just slightly out. So either people are making a lot of typos, or they're deliberately trying to slow down the opposition by getting them to fill in the wrong order to your New England biomedicine, or whoever's doing your New England nuclear, whoever's making your oligos for you. And there have been some concerns about research farms in some parts of the world where they're publishing a large number of almost identical papers, apparently looking at the effects of different genes in different places. Now, I'm sure that some of that's true. I don't think it's particularly widespread. In any case, in that graph of mine, even if you could make everybody improve by only, where's my graph of mine? If you could get everyone to improve by only, that didn't work, did it? If everyone here could improve by only 1%, like that, that's a 2% improvement. Okay, so everyone improves by 2%. The increase in value of research is the same as getting rid of the 20% most extreme research practices. So it's not that, of course, it's not that we shouldn't do this. We should do this, and the punishments for willful fraudulence should be high. But actually, we shouldn't focus too much on that, because actually what gets us more good is by everyone being a little bit better in what they're able to do.
Yes. Anyone else? So yeah, well, I think I do agree with most of. I mean, we do agree with a lot of things. I'll probably rehearse your speech on ALS and use it in my own lectures on publication. I quite like the analogy, and so yeah, and well, thank you very much for being with us. I mean, we realize it's quite late in Scotland, so you're probably
all right.
Yeah,
it's all right. My wife is busy watching Game of Thrones. I don't watch Game of Thrones, so she's grateful for the opportunity to watch it with her when I'm busy doing something else. So she sends her thanks also for the invitation, but thank you.
Okay.
And well, I'll see you soon, I hope. Okay. Right,
yeah, thanks a lot. Hopefully we'll help to answer some of our questions and build better data on how to assess research.
Yeah, if anyone wants to email me any questions coming out of this or anything, please feel free to do so.
Yeah, so can I send them all your email here?
Yeah, absolutely. Absolutely.
Okay, cool.
Absolutely.
Sure.
Okay.
Thanks a lot.
Thanks very much. Cheers, everyone. Bye.
Bye.